In this talk, I wish to discuss our laboratory methods for detection of CAR T cells and for monitoring its persistence after infusion. Right, so first of all, the objectives of laboratory monitoring after CAR T cell therapy include the following three aspects. First is the detection of minimal or measurable residual disease. That means whether there are residual cancer cells, be it leukemia cells or myeloma cells after therapy. Second is to document the immune reconstitution of the patient after immunosuppression. Say, for example, what is the lymphocyte subset numbers, lymphocyte function, immunoglobulin levels, etc. And then thirdly is to monitor for the CAR T cell persistence, which is the focus of this talk. In our lab at Hong Kong St. Thomas Hospital, we developed two methods for CAR T cell monitoring. The first is by flow cytometry, shown on the left-hand side panel. So basically, we are using monoclonal antibodies tagged with fluorochromes to detect antigen expression on cells that flow through the laser in a single file. And hence, the CAR T cells, for example, are detected by specific antigen expression. Second is a new method we developed is Droplet Digital Pcr. So I think in short, it's basically a special type of PCR technique where we compartmentalize the DNA molecules into droplets, for example, so that to enable the rare events to stand out and be detected. And this is applicable for very low numbers of CAR T cells existing in the body after therapy. Right, so first of all, flow cytometry. So this cartoon shows the T cell, which is engineered to express the chimeric antigen receptor consisting of the antigen binding domain. So this targets the CD19, for example, as in Kymriah, together with a hinge, the co-stimulatory domain, as mentioned by Professor Liang, and a signaling domain. So basically, this part will recognize the CD19. And for this reagent, this is a special type of biotinated fusion protein of CD19, extracellular domain, and specifically mutated human Ig1 Fc region to reduce the noise. So what happens is that we can detect the CAR T cells both by the anti-biotin fluorochrome, because this protein is biotinated, and also by the conventional anti-CD19 antibody. Okay, so there are basically two angles where we can detect the CAR T cells by this reagent. Right, so this is a plot of a study of the peripheral blood of the patient day 2 post-infusion. So at that time, we still do not have the biotin reagent, so we rely on the CD19. So on the left-hand side, it shows you the y-axis is the size scatter and the x-axis is CD45. So the lymphocytes are gated, in which, as you can see on the right panel, there are co-expression of CD19 and CD3 in 18.5% of the gated lymphocytes. So this is impossible in a normal situation, because in a normal lymphocyte, you cannot co-express both the B cell marker CD19 and a T cell marker CD3. So these cells are interpreted as the CAR T cells on day 2. Right, so we have a chance to look at peripheral blood by flow at 3 more time intervals. So at 3 months, at 6 months, at 7 months. Okay, so at that time, we already have the biotin reagent. So shown here, the y-axis is the biotin reagent. X-axis is the CD3, okay? So these green dots are the normal T cells, CD3 positive. And the CD3 positive and the biotin expressing rare events are interpreted as a CAR T cell. So as you can see, at 6 months, there are very few, only 4 events, and at 7 months, 7 events, okay? So I think when we plot the data, it shows that the CAR T cells drop to very low levels after 3 months and then persists at very, very low levels in the peripheral blood at 6 months and 7 months. But however, this reagent is not properly validated for the limit of detection. So in order to be more accurate in the CAR T cell detection, we developed a digital droplet PCR, ddPCR in short. So what happens is that we, first of all, to detect the CAR T, we target the TSG LLC region, okay, which is the junction of the co-stimulatory and the signaling domain. So this is specific for CAR T cell. And then it is targeted by a FAM labeled probe. Okay, we also target another gene region, which is a housekeeping gene, the CDKN1A, okay, and then targeted by a probe labeled with another fulcrum, the HKSH, okay, so as an internal control for normalization of the copy number. So we do this in separate droplets in different wells, so in a single plex fashion. And this is based on another real-time PCR paper from Davis et al. But mind you, ours is a ddPCR, but we modified from a quantitative real-time PCR to a ddPCR method. And then we're also fortunate to be able to get hold of some positive control cells in that we use the empty bag that post-infusion, and then we rinse it with a saline and then extract the DNA to serve as, first of all, to validate the assay, and then serve as a positive control in each run. And we're able to validate the limit of detection. So for the CAR T cells, by zero dilution, the linearity is from 100% down to 0.125%. And the limit of detection is 15 copies per 20 microliter reaction, or roughly 12 FAM-positive droplets. And the limit of blank is 0 copies, so there is absolutely no noise in this particular assay. The linearity of this housekeeping target, the CDKN1A, the linearity is from 100% down to 0.25%. But we are not too worried about the CDKN1A because this is usually occur in abundant numbers. It's only that the CAR T TSG LLC is a rare event. Okay, so I'm showing you the peripheral blood at 3 months. Okay, and the left-hand side is the CAR T, the right-hand side is the housekeeping gene. Okay, so the y-axis is the fluorescent signal, and the x-axis is the wells. So I think each well contains 20,000 droplets. Okay, and these blue dots, any dots above the purple line are the positive signal. So these are positive control. And for this patient at 3 months, there are actually detectable CAR T cells by ddPCR in the peripheral blood. Okay, and the right-hand side panel shows the housekeeping gene, which is detectable in abundant numbers. However, the bone marrow sample at 3 months, okay, shows no detectable CAR T cells. Okay, so at 3 months, CAR T cells are detectable only in the peripheral blood, but not in the bone marrow. And I'm showing you this species table, basically showing the trend of the CAR T cells throughout the half a year after treatment. So this column shows the CAR T cell copy number. This is the housekeeping gene copy number. And the CAR T cell over the housekeeping will give you a ratio and express as a percentage. Okay, so infusion pad contains numerous CAR T cells. And then before transfusion, none in the patients. But after infusion, you can see that the CAR T cells reach a peak at 154 copies at day 4. So note that the date of infusion is 18th of March. And then it slowly drops off, okay? And I've just shown you the data at 3 months, which is this set. So the CAR T cells detectable in the peripheral blood, but not in the bone marrow. So we look at the peripheral again at at 6 months and still detectable in peripheral blood. And at 7 months, it is barely detectable in peripheral blood, but not in the bone marrow. So the same situation as in 3 months, okay? But now it drops to very low numbers. And if you plot it, and the y-axis is the ratio percentage against time, you can see that, in fact, CAR T cells are detectable at 6 months, okay, but only barely at 7 months. So I can say that the CAR T cells detectable period in the patients, in this particular patient, is around 6 months. And then unfortunately, the lymphoma relapsed at around 7 months. So based on this experience of this patient, we conclude that documentation of CAR T cell persistence is feasible by both flow cytometry and ddPCR. And peripheral blood is the preferred sample, okay, because in the later time periods, it's detectable in the peripheral blood, but not in the bone marrow. And then disappearance of the CAR T cells may herald disease relapse. I wish to acknowledge Dr. Becker Yip for the ddPCR work, Sharon for the flow cytometry, and Professor Raymond Lang for allowing us to study the patient. Thank you very much.